Today we are going to read about another topic in the immunology lymphoid structure, that is spleen. So the spleen is located in the left upper quadrant of the abdomen and it is anterolateral to the left kidney. The spleen is protected by the 9th to 11th ribs. So the spleen is similar to the lymph node and it is encapsulated lymphatic organ. The lymph node filters the lymph while the blood is filtered through the spleen. So the blood is monitored immunologically by the spleen. So in human, spleen is a single large organ present in the upper abdomen. As I have already stated, it is present in the left upper quadrant of the abdomen and it is covered by the peritoneum. So you can see the cross section of the spleen, which shows the substance of the spleen arranged in the form of white pulp and the red pulp. So in the fresh section of the spleen, which is unstained, or we can also view the spleen in stained from hematoxylin and eosinophil. So the white pulp is surrounded by the red pulp. So in hematoxylin strained section, the white pulp appears basophilic due to the presence of the small lymphocytes, whereas the red pulp appears eosinophilic because of the blood sinuses that is filled with the RBC. The main function of the spleen is the splenic macrophage that is present in the spleen removes the encapsulated bacteria. So the person who have splenic dysfunction are very susceptible to the encapsulated organisms. So I want to focus on the clinical aspect of the spleen. So whenever there is splenic dysfunction like post-splenectomy state or sickle cell disease, or if there is any disorder of the spleen, then there will be the decrease of the immunoglobulin M. And with this decreased amount of the immunoglobulin M, there will be the decreased complement activation. The detail about the complement and the complement activation we will discuss in the later topic. But you should know that if there is splenic dysfunction, then there will be the decreased amount of immunoglobulin M, and this will lead to the decreased complement activation, which will ultimately leads to decreased C3b opsonization. And if there is decreased C3b opsonization, and then there will be the increased susceptibility to the encapsulated organism. So now we will discuss detail about the opsonization also later. So the post-splenectomy blood findings will be the Howell-Jolly bodies, the target cells, the thrombocytosis and lymphocytosis. So if there is any splenic dysfunction in the person, then the vaccinated is the must against the encapsulated organism like the pneumococci, Haemophilus influenza B and meningococci. I want to discuss a little more about the red pulp and the white pulp and the sinusoids, or we also called as the blood sinuses, which are filled with the RBC that is present in the red pulp. So sinusoids are the long vascular channels in the red pulp. So it is the barrel hoop, and the T cell are found in the periarterial lymphatic sheath within the white pulp, and the B cells are found in the follicles within the white pulp. The marginal zone in between the red pulp and the white pulp contains the macrophage and the specialized B cells, and it is where the antigen presenting cell capture the blood-borne antigen for the recognition by the lymphocytes. So this you should know that the basically the splenic macrophage, the spleen is the organ that monitors the blood immunogenically, and if there is any disorder of the spleen, or if there is trauma and the splenectomy is done, then there is increased susceptibility of the person to the encapsulated organism, because the splenic macrophages basically remove the encapsulated bacteria. So I hope you understood the topic on the spleen. So here in the spleen we talked about the histological structures, the various function of the spleen, and we will also talk about some clinical aspect of the spleen. So thank you for today. This concludes our topic on the spleen.